We're so pleased today to have Max Spurrell here with us. He's an MD PhD candidate at the Department of Immunobiology at Yale School of Medicine. His mentors are Kevin Herold and John Tsang. And the title of his talk is A Surviving Beta Cell Subpopulation Enriched in Patients with Type 1 Diabetes. A little bit about him. As I said, he is in the Department of Immunobiology at Yale School of Medicine. He completed his undergraduate studies in Brown before joining the laboratory of Dr. Nir Hacohen at Mass General, where he studied the spatial organization of the tumor microenvironment in human lungs cancer or how it shapes responses to immunotherapy. So that foundation in computational and spatial biology of immune tissue interactions now informs his work at Yale, where he's redirected his attention from cancer immunology to the immunology of the pancreatic islet, and specifically to the question of why a small number of beta cells survive for years and even decades in a tissue under active immune assault. We also just want to do a little bit of a shout out to his mentors. Kevin Herold's one of the preeminent figures in T1D immunology and beta cell biology. He's known for his foundational work developing and clinically validating teplizumab, the first FDA-approved drug to delay T1D onset. And then John Tsang is a systems immunologist with deep expertise in single-cell multiomics and gene regulatory networks inference, and he directs the Yale Center for Systems and Engineering Immunology. So we are very excited to learn more about the big question: what is the transcriptional state of beta cells that are still alive in a patient that has had type 1 diabetes for years, and what is it about them that made them survive? Thanks for joining us, Max, and we're really excited to hear this talk.
Cool. Thanks for the very kind introduction. So I'll go ahead and get started. So, as we all well know in this audience, type 1 diabetes is caused by the loss of insulin-producing beta cells. Beta cells are endocrine cells that live in the pancreas and make insulin. And and in type 1 diabetes, these cells are targeted and destroyed primarily by CD8 T cells, though other immune cells play a role as well. And importantly, because beta cells, especially in humans, aren't really thought to proliferate much, once a kind of critical mass of beta cells are lost, this causes lifelong insulin deficiency. And so this plot shows kind of beta cell loss over time. By the time an individual actually presents with symptoms of type 1 diabetes, that's stage 3 T1D, the vast majority of their beta cells have already been destroyed. So usually somewhere between 60 and 90% of the original mass is already lost. However, you'll also see that in this kind of schematic that there's an asymptote. So the loss of beta cells is incomplete and it doesn't reach zero. And so that's drawn schematically here, and that's built off of studies like this one. A couple really interesting studies from the Joslin medalist cohort. So these are patients who had 50 years, at least 5 decades of type 1 diabetes. And in this study, there were hundreds of individuals. And so this 2010 paper looked at these medalists that had 5 decades of T1D and did random C-peptide tests. And what they were able to find is that about 2/3 of the patient had detectable C-peptide, which is a marker of endogenous insulin production. And what that suggests is that there's still some beta cells or insulin-producing cells left in these individuals after 5 decades. And as many know, unfortunately, we can't get tissue access easily in the field of type 1 diabetes. So almost all of our tissue has to come from recently deceased cadaveric organ donors. And so in a follow-up study, looking at Joslin medalists, looking at their tissue after some of these individuals unfortunately passed away, when this follow-up study stained for beta cells, so that's shown in red in these images, that's insulin, they were able to identify beta cells actually in all 68 donors that they looked at. And so together, these studies kind of, I think, pretty convincingly show that persisting beta cells are both a stable feature of human T1D, so they can live for an individual's entire lifespan. And moreover, they're very common. So this isn't like a one-off thing specific to certain individuals. It's probably most, or if not, it seems like all individuals have some surviving beta cells that are not destroyed. And almost a decade ago now, the Herald Lab had done this study in the NOD mouse, looking at different populations of beta cells in the NOD mouse model. And what they did is they used, so these flow plots show beta cells, and they defined 2 populations that differed based on granularity, so that's the side scatter. And they found that these bottom cells increased over time coincident with the onset of insulitis, but that this only occurred in the NOD mice and that this didn't occur in B6 mice, which don't get diabetes. And then when these bottom cells were sorted out and compared to top cells, which are kind of the normal population, at least in B6 mice, they found that the bottom cells were kind of more resistant to immune-mediated insults. So say immune infiltrates, purified infiltrates that were co-cultured, or inflammatory cytokines, which are known to induce cell death, were kind of less effective at killing bottom cells compared to the top cells, which were more susceptible to cell death. So that kind of led to this really nice story where there's this population of resistant beta cells that emerges in the NOD mouse model, and it is kind of more resilient to immune insults. And so both that NOD paper and the human data that we've observed have kind of pointed to this phenomenon where there's certain beta cells that are able to escape autoimmune killing. And a really, really, I think, important and interesting question is like, what makes these beta cells special? And why are they able to avoid killing compared to the vast majority of other beta cells that are targeted and destroyed by primed autoreactive T cells? And translation, I think this is a potentially really important question, because if we have evidence that there's a naturally surviving population. So if we can identify important features that's helping these cells survive, we could potentially leverage that information to kind of engineer immune-resistant beta cells, which is a really kind of hot topic in the field right now. So our kind of high-level approach, and we'll go through some of this in more detail in the subsequent slides, is we reanalyze the HPAP data set, which is I think by far the most extensive T1D islet single-cell resource that we have right now. And it includes both individuals with established T1D, individuals who are autoantibody positive, who maybe they're in stage 1 or 2, but we don't know what eventually would happen to them, but they might be in the early stages of disease. And then non-diabetic individuals. And our strategy to ask the question, are there specific beta cell types that are enriched in those beta cells, programs, phenotypes, states, anything like that, that are enriched in those beta cells that have definitionally survived in patients with type 1 diabetes? And so our strategy was to first kind of computationally sort out the beta cells. And then we did this gene regulatory network inference-based clustering method, which I'll kind of walk us through in the next few slides. So just some information about the cohort. So there's 12 donors with type 1 diabetes that's currently available on HPAP. The disease duration ranges from 0 to 10 years. Unfortunately, sometimes the disease duration is also missing. And kind of importantly, we were able to identify surviving beta cells in 10 out of the 12 donors. So this plot shows beta cells as a fraction of endocrine cells. And, reassuringly, and as expected, the blue bars tend to be a lot lower than the red bars. So there's fewer beta cells in type 1 diabetes than in non-diabetic individuals, which is what you'd expect. But importantly, that number isn't 0. So we still have a few thousand beta cells that we're able to recover from patients with type 1 diabetes across most of the donors. And so this kind of lets us ask the question or interrogate these cells. And one really important thing that we noted early on is that there is unfortunately a lot of technical noise in the data. And I think a lot of that is intrinsic just to the difficulty of generating it. I would imagine, and also with any like dissociation-based assay like 10x, there's a lot of like transcripts that get like into the supernatant, and that causes a lot of background noise, which is a known issue. And so what I'm showing here is a UMAP of all the cell types in the islets without any integration methods that are conventionally used. And what you can see in the beta cells that are marked by insulin is that the differences in 10x kit chemistries kind of drive the different, I don't want to say populations, but they drive the beta cell position in the latent space. And kind of a key issue, though, is what is noise versus what is a real biological difference is often a gray area. And so traditionally, we have all these methods like Harmony, for example, that are integration methods that seek to kind of fix this phenomenon. But in general, a lot of these methods tend to overcorrect and kind of favor correcting technical noise and potentially losing interesting biological signal. And I think this is one of the reasons that maybe different subtypes of beta cells haven't been identified yet in type 1 diabetes. So our approach was to use a gene regulatory network. It was to infer a gene regulatory network and then use these scores for subsequent analysis. So just briefly, the way this works is you input a cell by gene matrix that you get, as part of any single cell analysis. And then this method, scenic, which is pretty widely used, searches for transcription factor gene correlations, which is shown here. And then to infer direct regulation, it actually looks at the kind of enhancer sites that might be proximal to that gene. And if there's an enhancer site for the transcription factor nearby the promoter of a given gene, that suggests that there might be direct regulation. And so then you can get a list of genes for each transcription factor across the whole data set that it's thought to regulate. And that can be scored on a single cell level. So you get after you put in a cell by gene matrix, you get a cell by transcription factor activity matrix. And then you can use that as kind of an input for more traditional dimensionality reduction and clustering. So that's what we did. All these cells here are beta cells. And what you can see importantly is that these are distributed across patients. So these different these are each bar is a patient, and you can see that the different colors aren't necessarily specific to individual patients. And when we look at this, again, it seemed like the different kit chemistries might have been driving some of the artifact. When we split by different 10x kit chemistries, we see that a lot of the kind of technical variation seems to no longer be an issue. It's hard to know exactly why this is, though kind of hypothesized that this uses a diffusion model, which inherently adds noise to the data, and then learns to denoise the data. So it might be more robust to data to noise. And then also it's a rank-based method. So those are also known to be a little bit more kind of resistant to noise. And this is the critical finding of this approach. So again, these are all beta cells, and we identified different populations, and we were able to identify one population of beta cells, which was cluster 3, which is this kind of magenta population at the center of the UMAP that was strongly enriched among the beta cells from patients with type 1 diabetes. So these plots, each dot is a patient, and the fractional composition of beta cells assigned to that cluster over all beta cells in each patient is shown. And what you can see is that for cluster 3, the median kind of frequency of cluster 3 beta cells was about 15% in patients with type 1 diabetes compared to non-diabetic or autoantibody positive patients, where it was like 3 to 4%. And so what this means is we've kind of used this approach to define a population of beta cells that is enriched among those surviving beta cells in patients that already have type 1 diabetes. Okay, so next we looked at which transcription factors were defining the activity of cluster 3. And so these are kind of the top 6 that we focus on. And as you can see, they're strongly enriched in cluster 3 beta cells compared to non-cluster 3 beta cells. And what was kind of really interesting and exciting is when you do a literature search for many of these transcription factors, there's actually a ton of mechanistic studies, just a subset are shown here, for several of these transcription factors that support some kind of pro-survival or immunomodulatory role in beta cells in an immune context. So for example, there's several papers from the Azaric lab, for example, that show that IRF1 has really kind of important roles promoting beta cell survival. And similar papers can also be found for several of these other transcription factors. So this is kind of encouraging because it kind of, or I don't want to say encouraging. This suggests that this program might actually be a beta cell survival program. So then we looked at before I showed transcription factor activity scores, then we look at the transcripts. This is kind of more traditional, like single cell analysis. So these are what transcripts are enriched in different clusters. And we noted that there are several transcription factors, as expected, whose transcripts are enriched in cluster 3. So all the kind of TF activity scores that define cluster 3 are also enriched in cluster 3, which is a good kind of internal control. We also saw that several other transcription factors were enriched in cluster 3. For example, IKZF4, which is a known T1D risk gene, and ELF3, which is another transcription factor that has been shown to have some pro-survival role in beta cells. And then it was really interesting to see that there were several immunomodulatory genes that were enriched in this population. So SOX1 and SOX3, which are negative regulators of cytokine signaling, HLA-E, which can inhibit NK and T cells, and then CD47, which is the don't eat me signal that is currently being used in the SANA kind of platform. And then we also saw another kind of group of genes that we saw related to stress or DNA damage. So these GAD genes and DDIT4 are known like DNA damage response genes, and then CDKN1A, which encodes P21, which can be associated with senescence as well.
Max, we just had a question in the chat. I'm just going to stop you for a second. From
Oh,
Pierre
sure.
Medico, are these surviving beta cells in typical islets or as a single or beta cell clusters? I just wanted to see if you could.
Oh, sure. So this is dissociative single cell. This is a limitation of this approach that I was, that's kind of would be an interesting future direction, but it's dissociation based. So to generate, drop-seq based 10x data, we lose the spatial coordinates, which is a very interesting kind of future direction, but can't be addressed. It's like a very common limitation to these approaches.
Thanks for qualifying.
Yeah. So another kind of transcriptional phenotype that we identify in these C3 beta cells is evidence of dedifferentiation. So we see some of like the lineage specific TFs downregulated, such as MAFA, MAFB, and NKX61. We also see decreased expression of several genes that are related to insulin processing and secretion, some of which are also autoantigens. So it's interesting to highlight, for example, PCSK1 and carboxypeptidase E. So these are genes that are involved in proinsulin processing. And as we know, an elevated proinsulin to insulin ratio is a feature of type 1 diabetes. So it's interesting to speculate that potentially having more C3 beta cells would lead to an elevated proinsulin to insulin ratio. And then also dedifferentiation has kind of been hypothesized to be a way that beta cells could protect themselves because certain autoantigens, which are also functional proteins in beta cells, can be downregulated. So we see some autoantigens are downregulated, but others, for example, insulin is not downregulated. And by decreasing the amount of antigen, beta cells are hypothetically making themselves less visible to autoreactive T cells, which recognize those antigens. And so we see this kind of potential dedifferentiation both on the transcriptional level and then on the pathway level. So there's this kind of strong, significant downregulation of the widely used beta cell pathway. We next kind of wanted to ask what might be driving the formation of these cells. And one feature that kind of stuck out to us is that there was a lot of evidence of cytokine signaling in cluster 3 beta cells. So for example, there was a very strong signal for both interferon signaling and then NF-kappa B signaling which was enriched in cluster 3. And kind of on that note, the phenotype, though, especially for interferon, turns out to be a little interesting. So it's not cluster 3 beta cells express some ISGs or interferon stimulated genes, but it's not, as far as we can tell, like a classic obvious interferon signal. So it seems like they express a subset of them. So to illustrate this, we like class 1 hyperexpression is a well-described hallmark of type 1 diabetes islets or of type 1 diabetes beta cells. But what we saw is that actually like canonical class 1 molecules like HLA A, B, and C and beta 2 microglobulin aren't really any different between cluster 3 and non-cluster 3 beta cells. However, HLA-E seemed to be enriched, so it seems more specific to HLA-E. And then also we saw that there were many ISGs that were enriched in cluster 3, but they were generally lowly expressed. Like, so this is the percentage or proportion of cells expressing these genes. And as you can see, they're really only the cells that express these genes, they're all in cluster 3, but it's a minority of the cells. And then there's other ISGs like CXCL10, for example, that are usually robustly induced by interferon that we just don't see at all. So it seems to be kind of a subset of ISGs that are expressed. So again, we wanted to ask the question. So then we wanted to like think about this orthogonally, what stimuli might be driving C3 formation. And so our approach was to leverage the Maestas et al. Paper from Jeff Millman's group. And what this group did is they took primary islets from non-diabetic donors, and then they treated them with various stimuli to induce stress. And so these included both ER stress inducers as well as cytokines, either alone or in combination, and then they did single-cell RNA sequencing. And so our approach was to use this data, kind of do initial processing to identify the beta cells, and then check which stimuli produce the signal that's most like the C3 phenotype that we observe in vivo. And so we did this, and just to walk you through the method a little bit, so we took the top down and upregulated genes in cluster 3, and then each dot here is a patient across different conditions. And then we checked how beta cells in aggregate scored for these different gene sets. And so what we saw is that inflammatory cytokines seem to reproduce the changes that we see in vivo. So for example, this is control beta cells. This cytokine mix of interferon, TNF, and IL-1 causes downregulation compared to control of the cluster 3 down genes and upregulation of the cluster 3 up genes. But also what was interesting is it turns out that each cytokine individually seems to be able to do this. It's most prominent in combination, but these are actually all significant unless indicated by NS. So what you can see is interferon, IL-1, and TNF can all turn down the genes that are downregulated in cluster 3 and are able to upregulate the genes upregulated in cluster 3. And so that's kind of broadly, and then we can look at the actual individual transcripts that were kind of used to whose activity scores defined cluster 3. And we can see that treatment with the cytokine mix seems to potentially phenocopy what we saw in vivo. So for example, IRF1, BCL6, all these other transcription factors are strongly upregulated by this cytokine mix, and MAFA and MAFB are strongly downregulated. And so this suggests that cytokines seem to be driving this state in vivo. So at this point, kind of had two questions that we were really interested in. So one, is this phenotype a beta cell thing, or can we observe this in other cell types? And then also cytokines don't occur in a vacuum. So that's a cell extrinsic stimulus. So if they're there, we should be able to see evidence of this in other cells. And the frequency of C3 beta cells and cytokine signaling in other cells should correlate across patients. So to ask this, we basically repeated our entire approach now on pancreatic alpha cells. And so we redid our GRN inference and clustered on those module scores. And then we're able to identify, again, several populations of alpha cells. And when we did this, we were able to identify an alpha cell population, again, cluster 3, that's just a coincidence, that seemed remarkably similar to the beta cell cluster 3 that we previously identified. So for example, the same transcription factors were highly enriched in this population relative to the non-cluster 3, now alpha cells. And when we directly compare the differentially expressed genes by log fold change among beta cells and alpha cells, we can see that actually both cluster 3 populations upregulate the same set of genes and downregulate the same set of genes compared to their non-cluster 3 counterparts. And so what this suggests is that this cluster 3 population is kind of it's probably the same program, and that it can be turned on in both alpha and beta cells. And then the last thing that we wanted to examine is if their frequencies are correlated, which we expect that they would be, and it turns out they are. So if they're cytokines, they should be acting on both cell populations. And what we see is that when cluster 3 beta cells are very low, there's a concordant very low amount of cluster 3 alpha cells kind of highlighted here. And as soon as it gets above 0, when one population rises, the other does as well. So there's a strongly significant Spearman correlation. So overall, we use this GRN inference-based method to identify kind of a novel population among those surviving beta cells in patients with type 1 diabetes. There's a lot of really interesting literature that can kind of be tied together by the program that's expressed by these cells. We think that inflammatory cytokines are driving the formation of these cells, and we don't think that this is necessarily specific to beta cells. And kind of big picture, we wonder if this might be like a natural cell survival program. And this remains to be tested, of course. But it's conceivable that if it is, that potentially augmenting parts of this program could be useful to engineer, say, immune-resistant beta cells. And this is a QR code to our preprint if you want to read more. And but I also want to address some limitations of this work. So of course, we're looking at cross-sectional data. So we can't establish causality and say definitely this is a cell survival program. We can just kind of hypothesize it based on the fact that these are cells that have survived to this point, and the literature supports that a lot of genes expressed by them are associated with survival. So I just want to make that clear. And it's possible that maybe they're not the cells that are resisting killing. Maybe they're those being killed. So this is impossible. We hypothesize it's this one, but this remains to be tested, and it's impossible to actually prove this, I guess, using cross-sectional data. And then lastly, as was mentioned before, we lose the spatial localization of these cells, and it'd be really interesting to see where these cells are, like if they're focal in certain islets or if they're more diffuse, because it would provide some clues kind of about what's driving them, and also help us resolve like what cell types might be producing these cytokines that prompt the formation of the phenotype. So that's it, I guess. And here are my acknowledgments. I'd like to thank both the Herald Lab and the CSCI and John Sang. And that's all I got. So happy to take any questions.
Thanks so much. Yeah, fantastic. I think it's really interesting to have some new tools heading back into some of these repositories that took so long to assemble and are kind of sitting there waiting for more analyses. And it's great to see you guys wading into this. Here's a couple of questions already, and if you have a question, please raise your hand and I'll unmute you. But from Klaus Kestner at Penn, and as we know, Klaus is heading up the HPAP initiative. Interesting that you find P21 in surviving beta cells. It has been suggested that elimination of senescent beta cells delays diabetes in NOD mice. Do you want to
Yeah,
comment?
it is interesting. I didn't see any other I think the senescence literature is interesting.
Mm,
I didn't want to use the word they are senescent beta cells because that's really the only classical. I mean, I'm not a senescence expert, but that's the only senescence gene that I really saw. And also that P21 gene is also an interferon stimulated gene. So if you just take, any cell type and put some interferon on them, they'll turn on P21. Um, based on some reading. So I think establishing senescence requires kind of a more, there's certain experiments like beta-gal staining, for example, that I think you'd have to do to actually show that it's senescence. But they do have that transcript. So I think leaving it at maybe is probably most appropriate.
Hope Peter Thompson's reading the paper and doing some thinking. Some Jacob Enriquez, have the alpha cell clusters been scored to see whether they are more related to top or bottom beta cells?
Yeah, so so we've looked at the top. There was RNA-seq done that on top and sort of top and bottom beta cells. And it seems like there's certain features that might be similar, but the actual genes could be different. So like that dedifferentiation was observed. I'm talking about cluster 3 beta cells versus bottom beta cells. Like dedifferentiation was observed in the bottom beta cells as well. Though it's actually interesting that certain interferon stimulated genes, I think we traditionally think of interferon as being and cytokines in general as being bad, but also that bottom cell population actually expressed interferon stimulated genes as well. So that's another potential similarity where it seems like seeing cytokines seems to induce this phenotype in both populations. But a lot of the actual one-to-one gene mappings aren't direct, and that could be, there's technical reasons, there's species reasons. Yeah.
Alrighty, here's Bart Roop from Leiden.
Yeah, I enjoyed your talk very much, Max. There was one thing that I noticed in your slide. You talked about beta cell state, and in your voiceover, you talked about different beta cells, and I think you should actually stick to the state thing because I'm not sure there are different beta cells, but one big distinction of beta cells of diabetes patients versus all the others is that these patients are treated with insulin, so those beta cells don't need to function. So they need not be dysfunctional, they just don't need to function because there is exogenous insulin. Could you comment on that?
Yeah, so I guess I'll comment on the first point first about state versus type. I agree with you. I think it's a state thing. So this wasn't we did this, we just never included it. A different way to instead of like traditional DEG testing where it's between two groups, we actually notice that if we go back here, we mentioned it a little bit in the preprint, but all these transcription factor activity scores are on the same principal component. So it's the same axis actually driving this variation. And if we just look at the genes that are most and least, like that have the strongest positive or negative correlation with this principal component, it actually turns out to be a very similar set of genes to the cluster 3 up and down genes. So I do agree with you. I think it's probably more likely continuous as opposed to discrete populations. I think just a lot of the tools are better for analyzing cells as if they were discrete, even if that's not biologically realistic. So I do agree with you. And then about your second point about exogenous insulin, I think a lot of it's hard to know actually. So I think a lot of, if you actually go through the clinical metadata, one reason that a lot of these individuals end up passing away, they tend to be younger. A lot of them come into the hospital in pretty severe DKA. So actually, I think that even though, yes, you would expect them to be, generally on insulin, I think a lot of these individuals who probably passed away, at least in the kind of acute phase weren't on any insulin because they were in DKA. So, but I think it's hard to. I think with 12 patients, you'll never be able to know that. So I
Well,
think it's interesting.
you might want to analyze that. You want to make a distinction between those that are and those that are not dysglycemic.
Yeah, yeah, they're dysglycemic, but then I think the problem becomes is they have very different hospital courses.
Oh yes, no,
Yeah,
that's the biggest confounder of all. So I'm glad you noticed that.
yeah, and obviously once they come in, they're started on insulin, but then that's not what kills them. So it's I think it's with 12 patients, you'll never, we can do
No, I agree. Yeah, thank you.
interesting kind of happenstances, but we can never say anything definitively.
I agree. Thank you, Max.
Yeah, to that point, thank you for that back and forth. That was great. To that point, Max, how might you kind of use this strategy to dive into data that is coming from those in stage, one, two, in the pre-diabetic zone, those who are on their way,
Yeah.
and pull out signal.
Yeah, so I think it's hard because again, like I think one big, a lot of like the cool immune studies coming out nowadays have time, like at the
Hmm.
systems level have time as an axis, and because we're reliant on cadavers, we can never really use time to understand how things change dynamically. So I think that's like a big limitation just in the field in general. So I don't know how,
Well, I mean, I guess you might think about cadaveric tissue that is from those who are at earlier stages, perhaps.
Yeah, so it's actually interesting. So just to note on this, I guess. So there's this cool paper from the Vahedi lab a couple years ago that used a machine learning classifier approach to try to. So they trained the classifier on cells from non-diabetic patients and cells from type 1 diabetes patients. And then they asked the classifier to classify the autoantibody positive patients as either diabetic or non-diabetic in a cell label agnostic kind of way. And they kind of picked out two patients that they thought were most likely to get type 1 diabetes based on their classifier. And it turns out to be the two autoantibody positive patients that have the highest frequency of cluster 3 beta cells.
Hmm.
So, I think that's kind of pretty
Yeah,
interesting.
it is.
But if this
Yeah.
could specifically be, but the problem is we'll never know ground truth because we don't know what happened to these people. So we can
Never say
say,
never. There could be other data that you don't know about yet.
yeah.
Just as an aside, another MD, PhD out of Yale, Zach Freiberg, just had a very cool paper June 9th, well, preprint. And they did an AI guided analysis of human pancreatic islet sociology, revealed distinct cell compositional changes in type 1 diabetes. So now they're coming at it, sort of the progression using AI. Very interesting. Maybe it would be fun to give him a call and exchange thoughts on some of your findings because I think you might have a great conversation. Kevin Harold, would you like to?
Yeah, actually, I had just a couple points. One is to Jacob's question about the alpha cell clusters and the top and bottom. Those original studies involve the MIP-GFP mice, so they probably were beta cells, the top and bottom. But the other thing I wanted to bring up is the interesting point that Klaus mentioned about P21 and the role of senescence and type 1 diabetes. I think the questions are not the same about the role of P21 here and in the senescent and in the NOD model, because what's happening in the NOD is sensitization
Mm,
of the immune response to kill that leads to killing. In this case, we're talking about survival of beta cells. And actually, P21 has been shown to reduce ER stress. This is in type 2 diabetes and improve beta cell function. So I think they're different, whether we're talking about immune sensitization and beta cell survival.
yeah, that's an important point. Thank you for sharing that. Al Powers from Vanderbilt.
Kevin, you said you had a couple of points. Did you want to make your other points?
Kevin.
I did. That
Oh, you
was
made all
two.
your points.
That
Okay.
was two.
Okay,
That's
he
two.
did
Okay,
too.
good, good. So Max, thank you very much. That was very interesting, very provocative. I use that in a positive way in terms of being provocative. So two comments. One is the changes that you see that correspond to cytokines. That's a nice correlation, but how comfortable are you that the islet's in vitro response to cytokine actually mimics what's going on in the human pancreas in T1D?
Mm.
Oh, yeah. That's a great point. So I think I was very deliberate to say it's cluster 3 like. I don't want to say it's the exact same thing. So as I mentioned before, if you put interferon on a beta cell in vitro, you're going to get a very, very robust, rapid induction of like a classic set of genes. And the point I was trying to make is you see some of these genes in the cluster 3 beta cells, but not all of them. And of course, there's several other technical factors that kind of make it hard to know. So one, obviously, in vitro, beta cells aren't doing their physiologic job of insulin secretion, which kind of, is difficult to replicate. And then also, another important thing is that when the HPAP samples are processed, maybe others know more about this than me, but the understanding is that they're cultured in vitro for several days prior to their actual sequencing. And so what this means is that certain immune cells that might be active in the pancreas, which may not be as tissue resident, can egress and leave. So when you actually look at the HPAP dataset, there's very few immune cells there. Though I would guess that those cells were there originally, they just probably left in vitro. So I think that there's kind of all these like technical factors that make it really hard to tell. But so the absence of a signal has to be interpreted carefully. However, the positive signal that we do see in terms of similarities, I think pretty convincingly, at least I'm pretty convinced that cytokines do play an important role, though it's impossible to say that they're solely responsible. And we don't know which cytokines exactly because it seems like they all can do this individually. So we don't know which cytokines are doing this, the timing. There's a lot of unknowns, but I think in my mind, it's pretty clear that cytokines play an important role.
Yeah, Klaus, who's the one whose group did the sequencing and cultured the cells. You want to comment on Max's uh, thoughts about culture and influence of that?
No, I mean, I'm certainly very happy that these data were reanalyzed. It's good that many people are using them. And, as I wrote in my note, I mean, I apologize to the field that because we are started in 2016 and we get now donors, months by months, week by week, we don't really get them. The companies keep changing the platforms that we're using, so we really have no choice, but because they don't manufacture the old version anymore. So that's why a lot of things, why the data are so noisy. So I'm glad that you've really found a way around that. And yes, this is not doing mouse experiment. I mean, I'm a mouse biologist. I used to do that and have 10 controls, 10 mutants. They were all the same age, same sex, whatever, and I have no control over any of that anymore. So we have to analyze what we get, but I think you were able to really distill a great deal of information out of this. So it's really congratulations to that.
And then Max, this is Powers again, could you speculate if you had unlimited money, unlimited time, what would be your next experiments based on the data that you have?
That's an wow, that's a great hypothetical.
You're going to be very ready for your dissertation defense after this call.
Yeah. Let's see. Well, I think there's a few interesting directions. So one is I think like, we present this method as a way around the noise, but I think ultimately like if there was less noise in the data, we wouldn't need this approach. So I think if there was unlimited sample, I mean it would be great to confirm this and correlate it with certain phenotypes like disease duration and like a larger cohort of patients where we don't have to use this method. And then I think the mechanistic work is very interesting. So, we have this set of genes that we see enriched in this population. And there's a lot of, like, as I mentioned before, there's a lot of literature supporting that this might be protective. But then I think the next step could be to actually test this in, like, say, overexpress some of these C3 enriched genes in beta cells. And I think the important thing is that, we see downregulation, like dedifferentiation as well, which is obviously in the setting of iPSC-derived beta cells undesirable. So if there's but of course we know the immune system is very nuanced, and so there's reason to believe that maybe we could uncouple the survival effect from the dedifferentiation effect. And so I think like experiments where we would overexpress some of these transcription factors or genes and then assess a potential survival advantage could both help us understand, whether this really is a survival program, like contextualize the in vivo findings, and then also potentially enhance some of the, efforts to make immune resistant stem cell derived beta cells. Because I think like my understanding is that a lot of the combination, like say like beta 2M, CIITA, knockout, CD47 overexpression, or PDL1 overexpression, like those are generated empirically just based on what we know about immunology. But if we can actually use the biology to guide us to make kind of a tougher beta cell, I think that could be an interesting approach.
Yeah, I think that it's so hard, but I think somehow we have to connect this data to sections of pancreas, right? Because that's what we have, right? I mean, in pod, whatever, right? So that's the struggle of how to do that because as you said, you don't know about the spatial distribution and this and that sort of stuff. And so I'm not sure that's a tough thing
Yeah.
to do to change, to identify changes in transcription factors in sections and things like
Yeah, I will note that there's a Decio Azeric paper, I want to say from 2018, that did immunofluorescence that co-stained for insulin and IRF1, which was kind of like the top hit in our analysis. And they found that IRF1 was focally, they could only detect it in islets of people with type 1 diabetes. So it was absent in non-diabetic individuals, which is highly consistent with what we saw. But they found that actually the IRF1 positive beta cells were focal. And so they were only enriched in certain islets and weren't kind of broadly distributed. So I think we have some initial, I think the spatial question is really important and really interesting, but I also think we have some initial hints that this is both kind of intuitively based on how cytokines work, and then also that study that this is probably a focal process.
I guess just to riff off of that IRF1 comment. So like if you think about the translational implication you're kind of talking about, you sort of say, maybe the C3 program might be a target for T1D prevention or reversal. Like if you think about IRF1 activity, if it's driving survival, would you want to induce the state in more beta cells early in the disease? And if so, like how might you think about doing that without compromising
Yeah.
the function given the, dedifferentiation signature?
Yeah. I mean, I think that, before I could speculate on how to do it, I think a lot more of the biology would need to be worked out. But I
Yeah.
think just like big picture,
Thought
like
experiment. Thought
the
experiment.
immune, I think the immune system, like say in cancer, like interferon can be um like obviously anti-tumor, but it can also chronic interferon can support tumor growth. And so in the immune system, it's so carefully tuned evolutionarily to do multiple balanced things at once that I think there's no reason to not expect that like the beneficial impacts of like say cytokine stimulation, which there's lots of papers about, couldn't be uncoupled from a lot of the negative impacts of cytokine stimulation. So, but it would have to be worked out. But I think a lot of, like there's probably multiple processes happening inside these cells, and the next steps would be to figure out like which ones, might we could leverage. But that's pretty hand wavy, to be honest.
Hmm. Well, I mean, have to start thinking sort of big before you start developing your experiments. So I think it's great. Again, I just want to compliment you on like going back into these HPAP data and pulling out something novel and really interesting. And we hope to see more from you. I hope you certainly stay in the field. It's amazing what you've done to date. Um, and I would also say that I did mention Zach Freiberg. He is going to be presenting at the think tank July 16th. And his paper also quite interesting as he uses AI tools to further sort of dissect pre-existing data. So thanks again, Max. Great job. And we'll keep an eye on your next publication or when the preprint comes to full papership.
Sounds good. Thank you so much for having me.
Thank you. Have a great rest of the day. Bye-bye.